Okay, DNA as a nucleic acid, of course, is built up of nucleotides. So we have what's called a DNA nucleotide, which is again is the traditional 5-carbon sugar here. This time the sugar is deoxyribose, that's a sugar. And you have, of course, the phosphate attached to number 5 carbon, and the base attached to the number 1 carbon. And in this case, the possible bases that you find in DNA are adenine, thymine, guanine, and cytosine. Okay, sometimes we refer to them as just A, T, G, C, the possible bases found in DNA. Now, the structure of DNA. The structure of DNA, which was discovered, I think, in 1953 by 4 researchers. Normally, we only hear of 2 of them to give them credit, but there are actually 4 people that were involved here. So you have Rosalind Franklin. She's a British scientist. Let me get his name straight here. Rosalind Franklin. I'm forgetting his names already. So we got Watson, James Watson. I think Crick. What is Crick's name? That's his first name. And one more guy from also from England. Oh dear. Okay. It's in your PowerPoint, so forgive me for not knowing this 4. I forgot these 4 names already. Okay, so four scientists were involved in discovering DNA, and what they found is that DNA is a double-stranded molecule. Okay, and it assumed a alpha helical structure, like a spiral staircase. Okay, DNA. So let's draw for you how DNA is put together. All right. So DNA is double strand. So let's draw one strand of DNA first. So here we go. Okay, so in this strand of DNA, we're going to have the base can be A here, here it can be T, here it can be C, here it can be G, and the sugar, of course, you know, the number 5 carbon is a phosphate group. So this is how you have your nucleotides, and then the nucleotides they link together by forming a bond between the phosphate group and the number 3 carbon, like this. So it's called a sugar phosphate backbone. The phosphate of one nucleotide links to the number 3 carbon of the next nucleotide. Okay, like that. And then on the opposite side, what was also discovered is that the opposite strand ran in a different orientation called an anti-parallel orientation. So let's see here. So here we have, on this side now, Again, the phosphates will link to the number 3 carbon, like so, to link one nucleotide to another. And when we draw it this way, we can orient the strand. So in this strand, since the number 3 carbon here is free, this end is called the 3' end, and here the phosphate on number 5 is unattached, so it's called the 5' end. Over here, this will be the 3' end. This is the 5' end. So the strands are like this. It's 5 strands are 5 to 3 orientation and 5 to 3, like this. This is how they're oriented. It's called anti-parallel orientation of the strands. Then they also discovered that the bases pair in a certain way, always. It's a law of nature, that the A's can only pair with T's, and the C's can only pair with G's. Not only that, there are a certain amount of hydrogen bonds between them. So between the A and the T, you have 2 hydrogen bonds. Between, again, 2. Between the G and the C, you have 3 hydrogen bonds. Okay, and that's how you build your DNA. Double stranded, linked together, each strand linked together by sugar phosphate bonds, and then both strands are joined by what's called complementary base pairing, where G pairs with C, A pairs with T, and you have 3 bonds between the G and C's, and 2 bonds between the A and T's. That is your double-stranded DNA. Okay. Let's go one step further. Now, all the DNA in your cell. Remember, in each cell has the same identical supply of chromosome. All several trillion cells in the human body have the same exact copy of DNA, the same exact amount of chromosomes. And so the amount of chromosomes per cell is called the genome. So the genome refers to the total DNA in each cell. And so each cell has the same identical genome, except of course for your sperm cells and oocytes. They have half the genome. And this genome in humans, our human genome, contains 3 billion pairs of nucleotides. So basically there are 6 billion nucleotides in the human genome, and these 6 billion nucleotides are in each cell, packed in each cell. Now, the portion of DNA that codes for a protein is called a gene. So the gene is sequence of DNA that codes for a protein. Okay. And there are about 20,000 genes in the entire human genome. And this only accounts for 2% of the genome. The rest of it, the remaining 98% of the genome, is non-coded. Does not code for a protein. It was once disparagingly referred to as junk DNA, probably still is, but it's really not junk. We have found over time that in this batch of DNA that is code for proteins, you can have controllers of genes. Regions of DNA that control the genes that are there in terms of when they are turned on, when they are turned off, how often, how fast they get transcribed. Okay, so some controllers are amongst this what's called junk DNA. Also, here that we have ancestral DNA, meaning DNA in our lineage, ancestral history that's no longer active in Homo sapiens, but were probably active in apes before us, or even before that, in the water mammals that gave life to terrestrial mammals. So it is our ancestral DNA that's there, no longer active, may become active again in another 1,000 years. And we also have viral DNA, meaning DNA that has been delivered into our genome by viral infections over our history. And once it gets infected by a virus, it gets pushed into your DNA and is passed down through your history, okay? You may also even have bacterial DNA as well. So it's just a lot of, we're really composite structures, especially in terms of virus and bacterial DNA. There's stuff in here that's not human, but we can't live without it. So we're really kind of a combination of human and viruses and bacteria. We're not really pure human per se. No such thing. We're mixtures of things. We're all nature combined. Okay, so we have, these are the things that you may find among the 98% of the remaining DNA. What else is there? So let's next look. Okay, let's pause there.